Hello, today we will discuss about the eukaryotic organisms. It is very clear that eukaryotic organisms are much more complex than the prokaryotic organisms, and they exhibit a larger amount of structural, functional, behavioral and a lot of differences. Now let us just take a look about a typical eukaryotic cells. This is a typical eukaryotic cell, and in this we will just have a look about the structures. This is the cell wall. The inner membrane is the cell membrane. These small structure, rounded structure are the chloroplast. Then we have mitochondria. We have ribosomes. We have smooth endoplasmic reticulum. Then we have a nucleus. We have a smaller structure inside the nucleus, that is the nucleolus. Then we have a rough endoplasmic reticulum, which is adjoined to the nucleus. Then we have this vacuole, which contains certain storage granules, and we have cytoplasm, which contains all these internal structures. Now let us discuss some of the fundamental structures of the important structures of eukaryotes. Now let us take a look at all the membranous structure which is present inside a eukaryotic cell. First of all is the plasma membrane. Plasma membrane is very similar to the cytoplasmic membrane in the prokaryotic cells. These are mainly applied or actually performing the ingestion of food via phagocytosis and penocytosis. Phagocytosis is the ingestion of solid food and penocytosis is the ingestion of liquid food materials. They also contains a large amount of sterols which do not have in the prokaryotic cells. Now second structure is the endoplasmic reticulum. Endoplasmic reticulum are the sac-like structures which have a cytoplasm outside and a sac-like lumen inside. These ramifications are supposed to increase the surface area of the endoplasmic reticulum for providing a larger surface for the protein synthesis and other processes. There are generally two types of endoplasmic reticulum, as we have seen here. It is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. In rough endoplasmic reticulum, we can see the ribosomes are studded over the endoplasmic reticulum, while in the smooth endoplasmic reticulum, they are actually smooth, they do not have any attached ribosomes in it. In rough endoplasmic reticulum, They are associated with the protein synthesis inside the cells, whereas the smooth endoplasmic reticulum, they are associated with the synthesis of phospholipids and sterols inside the cells. Next we have the Golgi apparatus. Golgi apparatus are the series of membranous structure which are originating from the endoplasmic reticulum, and they are supposed to perform the biosynthesis of certain polysaccharides such as the pectin as we obtain in the algae. The next structure comes the nuclear membrane. Nuclear membrane is the surrounding structure of the nucleus, and they are a site for the specific proteins which maintains the structure of the nucleus. And there are certain pores on the nuclear membrane which allows the transport of certain molecules from the cell and towards the cell. There is an entry, it performs the transport of mRNA from the cell and allows the input of RNA polymerase enzyme into the nucleus, which performs certain processes inside the cells. Next we have the vacuoles. Vacuoles are associated with the accumulation of certain storage granules and the certain intermediate metabolites and ions which are stored into the vacuoles during the certain processes which are performing inside the eukaryotic cells. Second is the lysosomes. Lysosomes are associated with the digestion or ingestion of the ingested food. They form via phagocytosis and via pinocytosis. When these foods are ingested, the lysosomes are supposed to digest the foods into smaller particles which can be further utilized by the cells. Next is the peroxisomes. The second is the mitochondria and the chloroplast. Both these structures have a lot of similarities among themselves, so we will discuss them together. Both the structures have a lot of things in common. First, both are concerned with the energy conversions. In mitochondria, we can see there is a site of oxidative phosphorylation, and whereas in this chloroplast there is photophosphorylations. Both the structures have two membranes. The outer one is permeable, whereas the inner one is impermeable. In mitochondria, the inner membrane has a lot of foldings to form a cristae. These structures which have the inner foldings, these are known as the cristae, and these are responsible for the respiratory chain enzyme reactions. Whereas in the chloroplast, the inner membrane foldings are known as the thylakoids, which stores certain pigments inside the cells. Last but not the least, both the organelles have their own DNA, mitochondria, transcription and the translation machinery. Now let us discuss the cytoplasm. In a fluid matrix inside the cell wall and the cell membrane, there contains a liquid matrix which contains a lot of enzymes, metabolites and various proteins like actin, myosin and other structural proteins. In the cytoplasm, there are actually all the organelles are suspended inside it, and when we come to the cytoplasmic ribosomes, these ribosomes have a sedimentation coefficient of ATS, which is S is termed as the Swedberg unit, and AT is the sedimentation coefficient, which are divided in 40S and 60S. Next, let's discuss the nucleus. Let's discuss the nucleus. Nucleus has a definite structural utility surrounded by a membrane. It contains many chromosomes and certain linear DNA molecules. It also contains nucleolus, which are the site of RNA synthesis, and it contains a lot of structural proteins known as the histones, which are not present in a prokaryotic cell, which is a very significant difference from the eukaryotic and the prokaryotic. These nucleus are involved in the reproduction processes also, that is the mitosis and the meiosis. Next we have the storage granules. These storage granules are the membrane bound organelles, or you can say a granule, which contains certain proteins or certain starch and certain lipid molecules which are bound by a membrane and are utilized under certain conditions. Now let us discuss the cell wall. In the eukaryotic cell wall, there is a lot of structural, functional and chemical differences other than the prokaryotes. If we consider different organisms and we discuss the cell walls, first of all we have the algae. Algae has microfibrils in the form of cell wall, and which are made up of certain polysaccharide molecules such as the pectin. And if we discuss about the cell wall structure of a fungi, we can say that the structural polymer of the fungal cell wall is the chitin, which is a beta 1,4 poly N-acetyl glucosamine. And when we discuss about the protozoa, protozoa have a variety of structural and chemical structures like the protein, cellulose, xylene, calcium, carbonate and silica, which are present inside the protozoal cell wall. That's all for today. Thank you.